So the subject of tonight's presentation is the mysterious red blood cell. Wait, what's so mysterious about red blood cells? We've known about them for more than 350 years. They've been exhaustively studied by researchers. We understand what they look like, what they're made of, how they're made, and what they do. What's so mysterious? Oh, here's a close-up of a bunch of them. We know that they are these tiny red concave discs in our blood that carry oxygen, which they pick up in our lungs. Our hearts transport them throughout our bodies via our arteries. Then, when our bodies extract the oxygen, the heart returns the depleted blood cells along with carbon dioxide back to the lungs via our veins, where we breathe in fresh oxygen and breathe out the carbon dioxide. Here's some fun facts concerning red blood cells. First, about 28 trillion of the 33 trillion cells in our bodies, a little over 80%, are red blood cells. Now, I realize this 33 trillion is not exactly true for everybody. I understand some people have as many as 40 trillion cells. The 80% figure of red blood cells is pretty accurate, however. Number two, each red blood cell contains about 270 million hemoglobin molecules. We'll be talking more about that in a little bit. Red blood cells only last about 100 to 120 days. Then they die and must be replaced. Every second, our bodies produce about 2 million red blood cells. From the start of the replenishment process, it takes 1 to 2 days for each red blood cell to mature. The old red blood cells are broken down into their component molecules and recycled. Red blood cells are red because of the heme. We'll talk more about the heme in a little bit. And they're scarlet when carrying oxygen, and otherwise kind of a dark red, and that's the blue that you see sometimes in veins. Almost all vertebrates have red blood cells, but only mammals expel their red blood cell nuclei. Scientists acknowledge there's a lot we don't understand about the process of expelling the nucleus and other organelles from the maturing red blood cells. But I'm getting a little ahead of myself. Here's an overview of what we'll be covering tonight. First, basic information about red blood cells, how they're replenished, some life-saving properties, apologetics considerations, and we'll sandwich in some of those mysteries that puzzle me. I plan to pause about halfway through my talk to take any questions or comments you might have, and we'll do that again at the end of the talk. The more I study biology, the more I am awed and astounded at what I see. The suggestions, clues, and evidences of design abound as I look more and more deeply into almost any biological subject. And if what I'm seeing is design, that implies there must be a designer. Many people are convinced that what we see is evidence of evolution, not design. They believe there's a continuum of life which began with a single-celled organism 3.8 billion years ago, and through the process of natural selection, evolved over time into ever more complex forms. I'm skeptical that evolution can explain so many apparent examples of design. Whether you have a background in biology or have been content just realizing that there are certain obvious differences between people, animals, plants, and bacteria, I hope that over the next 40 minutes or so, you will begin to see what I mean. I cannot offer you proof, but please join me as we look into the mysterious red blood cell. In order to exchange oxygen and carbon dioxide, red blood cells carrying carbon dioxide, which they have picked up as they pass through the blood vessels, must get through tiny capillaries in our lungs. There, the hemoglobin picks up oxygen molecules after dropping off their load of carbon dioxide. On the upper left of this diagram is an alveolus, tiny little sacs in our lungs where the gas exchanges occur. So you see blue capillaries at the top of the alveolus. Those are veins which have been depleted of oxygen and which carry carbon dioxide back to the lungs, while the red capillaries on the bottom and right side are arteries which have picked up oxygen from the alveolus and now will send it to the rest of our bodies. The diagram on the right is a close-up of one of the capillaries where it transitions from a vein into an artery. Blood flows from the top to the bottom in this picture. The vein transports carbon dioxide into the lungs via the blue deoxygenated red blood cell at the top and deposits it into the alveolus. Let me show you. So here's the blue red blood cell, and the blood's flowing down this way, and here it's dumping off its carbon dioxide, and here it's picking up its oxygen. The vein transports carbon dioxide into the lungs via the blue deoxygenated red blood cells at the top and deposits it into the alveolus. From there, we breathe it out of our lungs. And then, as I said, we'll see that the oxygen gas, or O2, which we breathed in, passes through the capillary wall and into a red blood cell. Although this diagram doesn't emphasize it, the diameter of the red blood cell is about 25% larger than the diameter of many of these capillaries. So the red blood cells need to deform a bit in order to squeeze their way through the capillaries. The squeeze through the capillaries makes the absorption of oxygen more efficient. Please don't think I'm saying that our bodies absorb all the oxygen we breathe in and that all the air we breathe out is carbon dioxide. Turns out that we only use about 4% of the oxygen we breathe in, and the air we breathe out only contains about 4% of carbon dioxide. This diagram also shows some structures on the outside of the capillaries, such as type 1 and type 2 pneumocytes, and an alveolar macrophage, but their actual functions are beyond the scope of this presentation, except for the macrophage, which we will talk about a little bit later. The oxygen is carried by a rather complex molecule called hemoglobin. We'll spend some time considering how our bodies construct these molecules. And yes, as I said, each red blood cell contains about 270 million hemoglobin molecules. That's kind of an astounding number, I think. Each red blood cell, as I said, contains about an astonishing 270 million hemoglobin molecules. Red blood cells have an average lifespan of 100 to 120 days, after which they, along with all 270 million hemoglobin molecules, get too old and must be replaced. And here's where the mysteries start. Aside from red blood cells, most of the cells in our bodies are self-reproducing. So when new cells are required, either in the process of growth or because the cells have reached their shelf life, they simply divide in a process called mitosis. So whereas before you had just one cell of any particular type, now you have two. But one of the enablers of the process of mitosis is the cell's nucleus, because one of the first steps in cell division is DNA replication. The entire DNA strand is duplicated in cell division. But red blood cells don't contain any DNA. In fact, they don't have a nucleus or any mitochondria or ribosomes, or any of the organelles contained in most other cells. We will see how red blood cells are replenished a little later, but for now, just realize that without a nucleus and the DNA it contains, when their lifespan is up, red blood cells can't just divide like most other cells. They will have to be constructed using another process. And the lack of all of these organelles, in addition to having a flexible membrane, helps explain why the red blood cell is a concave disc. By losing all these organelles, room is now available for the 270 million hemoglobin molecules contained in each red blood cell. But how could the red blood cell know it needed to lose its nucleus and all these organelles, an unusual and unorthodox process? This is the first mystery. We'll also have more to say about how and when all these organelles are replaced later. So how are replacement red blood cells constructed if they don't replicate themselves? The answer is through a complex process in the marrow of our bodies, the marrow of our large bones, I should say. The process is called erythropoiesis, and since that term is something of a tongue twister, I'm also calling this slide How to Make a Red Blood Cell. Let's take a look at how this is done. I'm going to show you quite a bit of detail to help make the case that this process looks very much to me like a quite complicated recipe. Recipes suggest design to me, and I think this whole process looks designed. Recipes are created by chefs. Designs need a designer. It doesn't make sense to me that a random, unguided mechanism like evolution could have put such an elegant process together in anything less than an infinite time frame, much less the few 100 million years available. Please don't get lost in the details. My point is just that this is a really elegant, complicated, and specified process. And pardon my lack of drawing skills, this picture is intended to represent a large bone with a cutaway showing the bone marrow where the red blood cells are formed. On the right of the diagram here is a blood vessel where the red blood cells are eventually destined and which provides some of the raw materials to make them. Obviously, if red blood cells only last for 3 to 4 months and they constitute about 80% of our 30 to 40 trillion cells, there must be a huge creation and disposal problem, and there is. Thankfully, there is a process by which the old red blood cells and their hemoglobin content are broken down and recycled into their basic components, which then get reused with only a small amount of actual waste, which our bodies then excrete. Otherwise, the disposal problem would be overwhelming. I'll have a little to say about the recycling process later, but for now, just realize that it's non-trivial. Replacement red blood cells are formed in the blood marrow from specialized stem cells called hematopoietic stem cells. That's these cells right here. Or some people just say blood stem cells, so we'll just say that. There are various different types of stem cells, but all stem cells have the capability of reproducing themselves. That's what this little arrow indicates here, which basically says that the stem cell can replace itself, as well as they produce a range of what are known as daughter cells, which are different from the stem cell. Notice that I said that the hematopoietic stem cells can reproduce themselves. This is really important because if there was only an initial source with such a huge replacement requirement, our bodies would soon run out of the starter hematopoietic cells in the process, and the whole process would soon break down. In addition to red blood cells, hematopoietic stem cells can form myeloid stem cells as well as lymphoid stem cells. So here's the lymphoid stem cells and here's the myeloid stem cells. And then the myeloid stem cells can produce other types of blood cells like monocytes, platelets, and granulocytes, in addition to erythroblasts, which are the precursors to the red blood cells. Let's take a moment to consider the mystery of how stem cells can produce different daughter cells. Expanding on my bone marrow diagram, here's a slightly more complex picture of how blood stem cells differentiate into myeloid and lymphoid stem cells, and how the myeloid and lymphoid stem cells then differentiate into all the different cells in the blood. Each of these cells has exactly the same DNA and genes as all of the others, Yet, all of these cells are distinctly different from each other, but somehow they all trace their origins back to the same blood stem cell. But let's go back even further in the formation of the various different cells in our bodies. As a new animal embryo forms after fertilization, a single cell is formed from the sperm and the egg. That single cell is a stem cell from which all the cells of the animal's body will grow. But like the blood stem cell, this single cell, also a stem cell, has the same DNA and therefore all the same genes as all of the daughter cells it will eventually divide into. Although scientists are beginning to get a pretty good idea of how the various gene products which form all these different cells are turned on and off and regulated, we don't really understand how the stem cells know how to differentiate into the nerve cells, the liver cells, the blood cells, the cardiac cells, or any of the other 200 plus different cell types in our bodies. And at an even more basic level, we don't understand how that single stem cell can eventually grow into the same organism as its parents. Or in other words, how it can form the body plan of its organism. Lest you think I am exaggerating this problem, consider the following quote. Consider for a moment development from the egg's perspective. What a job lies ahead of all those cell divisions and movements and of making tissue layer, segments, and body parts. Development proceeds in logical steps, but where are the instructions for each step? How is it that wide stripes are made before narrow ones, or some bones positioned before others? Or how is it that some bones are long and thin and others short and fat? How do the toolkit genes know where to act and when to shape the development of form? Where are the operating instructions for the toolkit? Sean B. Carroll is one of the founders and gurus of the discipline of evolutionary developmental biology, or EVO-DEVO. So if anyone should be an expert on the process of cell differentiation, Carroll could be considered that person. In this book, Endless Forms Most Beautiful, here's the answer Carroll gives to his own questions. In most animals' genomes, the genes that we see occupy just a small fraction of DNA. A much larger part of our DNA consists of sequences that are not part of the simple code for any gene and whose function cannot be deciphered simply by reading the sequence. This is the dark matter of the genome. Just as dark matter in the universe governs the behavior of visible bodies, the dark matter in our DNA controls where and when genes are used in development. If you study the topic of developmental biology, you'll read about families of genes and proteins which have been shown to influence the formation of the different cells and body parts. But again, every cell in our body contains the same DNA and genes. What is not so clear is how those genes are turned on and off in some cells, but not in others. Here, Carroll calls this the dark matter of the genome. In other words, he is admitting that we are still in the dark about how this differentiation process works. Here's what Carroll has to say about that. By virtue of controlling how toolkit genes are used, our DNA contains the instructions for making and patterning body parts. These instructions are embedded in the dark DNA as genetic switches. The anatomy of animal bodies is really encoded and built, piece by piece, stripe by stripe, bone by bone, by constellations of switches distributed all over the genome. Now, some of you may complain that when Sean Carroll wrote this book in 2005, we've had almost 20 years now since then, so surely we have come up with the answer to the dark matter of biology problem. Well, we really have not. I agree with Carroll that the control of genes is likely the key to cell differentiation and the specification of body plans, but I'm not as confident as he is about whether this information is actually contained within the DNA. Maybe it is, or maybe it's encoded in epigenetics, or in the cytoskeleton, or in glycans, or in something else. And even if he's right, that the answer is contained in the DNA, since every cell in our body contains the same DNA. How could one cell differentiate from another? I would not fall back on something like, we don't understand it, so God must have done it. I believe that some enterprising scientists will one day unravel this mystery, perhaps with a cell differentiation or body plan code and/or body plan code. But for now, Carroll's dark matter of DNA will have to suffice. For now, this is the second mystery. By the way, what Carroll is calling the dark matter of DNA, many other scientists have called junk DNA because they have assumed it has no purpose, though there was a study that was done in 2012 called the ENCODE project, which basically said that about 80% of DNA is actually functional. So returning to the red blood cell slide, please notice there are several additional substances. And let's see, let me get them back up here again. Like EPO or erythropoietin, which is shown at the bottom of the diagram, which stimulates the erythroblasts at the expense of the monocytes. So this would almost sound like an answer to my problem. So EPO goes in and the erythroblasts are stimulated at the expense of these other guys. But why is that? Why are the monocytes and platelets and granulocytes not stimulated as well? I can't answer that question. So yeah, EPO does definitely stimulate the erythroblasts, but why? I'm not sure. So in light of what I've just said about cell differentiation, I think a good question would be, as I said, why doesn't EPO also stimulate the monocytes? But try to remember that the monocytes, that is these guys here, because we'll have a bit more to say about them a little later. EPO is manufactured in the kidneys and it's delivered to the bone marrow, but too much or too little EPO would result in too many or too few red blood cells. So EPO itself is controlled by two other proteins, but for our purposes, just be aware that EPO doesn't act completely on its own. And then there's three other substances, iron, vitamin B12, and folic acid, which enter the bloodstream from the foods we eat. And these are vital to the recipe. The iron is inserted in the heme, but the recipe also requires vitamin B12 and folic acid. And all three of these come from our diet. But fortunately, a reasonably balanced diet contains enough of these ingredients to supply all the red blood cells we need. And I haven't mentioned it, and I wasn't planning to specifically mention it, but in talking about the recycling process, remember that all of this recycling is actually done in the bloodstream. So all of those recycled products get sent back into the bone marrow during the recycling process. And as the red blood cells are being formed from the myeloid stem cells and the erythroblasts, so too is hemoglobin. And that's what I'm trying to show with this red here, because you get a little bit of hemoglobin in the myeloid stem cells, more in the various different erythroblasts. And finally, by the time you get to the orthochromatic erythroblast, you have a lot of the hemoglobin. Though it's not all, apparently, still some of it is still forming by the time you get up to the reticulocytes. So here again is a single hemoglobin molecule, and each of these hemoglobin molecules consists of 4 heme molecules and 4 globin protein molecules. The heme molecules are the small green wiry structures, so that's 1, 2, 3, and 4. They're shown at two are shown at the top and 2 at the bottom of this hemoglobin molecule. The globin proteins are the red and blue helical structures, 2 red and 2 blue. Most of human hemoglobin contains 2 of 3 different globin proteins. You don't need to remember these. I'm just telling you the fact that it consists of HbA1, HbA2, these are known as the alpha chains, and HbB, which is known as the beta chain. 97% of human hemoglobin consists of 2 alpha and 2 beta proteins, and that's known as hemoglobin A, or HbA. Interestingly, the alpha genes are found on chromosome 16, and the beta genes on chromosome 11. But the globin genes manage to find each other even though they're so far apart, and to form hemoglobin. There are a few other globin proteins accounting for the remaining 3% of human hemoglobin, but for our purposes, the differences are really not important. Here's a stick diagram of one heme molecule. Notice this is not, I repeat, not a protein in itself. It's basically a molecule. Creation of heme molecules is a complex eight-step process requiring 8 different enzymes. And I'm going to go through those, and I don't expect, we're not going to be a quiz on this. I'm just trying to impress on you that this recipe is very complex. The heme molecules are built in the myeloid stem cells and erythroblast at the same time as the red blood cells are being built. The most important fact, however, is that heme contains iron, which is signified by the symbol Fe there in the center. Well, how does the iron get in there? Well, you can see it's bound to 4 nitrogen atoms in the center of the heme molecule. It requires a special enzyme to catalyze the insertion of the iron into the heme molecule, which we'll see in a minute. The iron binds the oxygen we breathe into our lungs. So it's essential that it's there, and it's essential that it's iron, because oxygen is transported in the bloodstream itself. So theoretically, we don't really need the heme and the iron, but the oxygen carrying capacity is increased by a factor of 70 by the iron binding to the heme. So if it was just for the blood and the air, the oxygen in our blood, except for the binding to the iron, we wouldn't have anywhere near enough. We'll see that the recipe to build the heme molecule and to insert the iron are pretty well understood. But I ask, how could an evolutionary, random, unguided, naturalistic process know that the heme structure containing iron would be essential to life? This is the third mystery. So anyway, let's go on. Heme is formed in the mitochondria and cytoplasm of the myeloid stem cells and erythroblasts in the 8-step recipe shown here. I'm sorry. I know this is a little overwhelming, but bear with me, please. The yellow figure on the left, that is this guy here, represents the intermembrane space of a single mitochondrion. There's several thousand mitochondria in each cell. So the mitochondrion is an organelle inside each of the erythroblast cells. The formation of heme starts up here with D-aminolevulinic acid. I won't bother giving you a picture of what it looks like. It's it's not really all that complex, but that's where it starts. Or DALA, and that's derived from succinyl coenzyme A, and it's catalyzed by the enzyme ALA synthetase with the amino acid glycine, which comes from up here. The DALA then passes into the cytoplasm of the cell through here to react with additional enzymes. I won't go through them all. And then eventually comes back into the intermembrane space. And at this point, the ferrochelatase adds the iron to the heme. And then we have the hemoglobin, which meets up with the globin chains. If you think this recipe looks pretty complicated, I agree with you. If you don't think it looks complicated, I don't think you get it. Here's a listing of the 8 ingredient proteins in the form of 8 enzymes. Well, if you look at the previous diagram, you'll see that they're all listed there. Each one of them is a special protein, and each protein requires its own gene, which must be transcribed into messenger RNA before the messenger RNA is translated into the enzyme. Again, the details are not important. I just want you to understand that these are very specific, different genes and proteins. It's interesting to note that this is just one of several biological processes which have been observed to make heme in various different organisms. So if you look in a different organism, you'll see a slightly different process. I have a book that shows 3 different processes, depending on the different organisms. As I said, the first step, the synthesis of aminolevulinic acid, or ALA, starts in the mitochondria. But now that the heme molecule is formed, let's turn to the rest of the hemoglobin molecule. Hemoglobin also needs 4 globin molecules. These are proteins produced when the DNA of their genes is transcribed into mRNA and then translated into the globin molecules in ribosomes. As I previously said, there are several different types of globin molecules which can be included in hemoglobin, but the most common one is known as hemoglobin A. So now we have 1 molecule of hemoglobin, but recall that each red blood cell has about 270 million hemoglobin molecules. It isn't important that you follow or even understand the details of what's going on. I'm just showing this level of detail to point out that this is a really complex recipe or process, which to me looks designed. We understand pretty well how the globin proteins are made in the process for constructing heme molecules. What is not understood very well is what process describes how the 4 heme molecules get inserted into the final hemoglobin molecule. We understand pretty well how genes specify proteins, but not how non-protein molecules like heme get inserted in protein structures. And this is the 4th mystery. Notice now the final step before the maturing red blood cell leaves the bone marrow and enters the bloodstream. At this point, the cell is called an orthochromatic erythroblast, and in this step, the nucleus is ejected. What? How in the world can that be? After this, the cell is passed into the bloodstream, where it is now known as a reticulocyte. Here's the reticulocytes, and they eventually get into what are now known as a red blood cell. This is the first one. No, I only show one. But in this stage, that is the orthochromatic erythroblast, not only the nucleus, but all the other organelles are ejected as well, including the mitochondria, the endoplasmic reticula, the Golgi apparatus, and all the ribosomes. The red blood cell is the only cell which ejects its nucleus and DNA plus all its other organelles. This is necessary to allow the red blood cell to hold all 270 million molecules of hemoglobin and yet still be small enough and flexible enough to fit through capillaries whose diameter is less than that of the red blood cell. This was the first mystery we considered earlier. In my opinion, this process too looks a lot more like design than something which is evolutionary, unplanned, and random. Well, what about the disposal and recycling processes? I said earlier I'd say something about it later. Notice in the above diagram that in addition to the erythroblasts, the myeloid stem cell produces monocytes. That's right here, so you see the monocytes. Well, what are monocytes? Here's a picture of one. I couldn't resist showing you this picture because I love it, although I acknowledge that it doesn't really tell you much about what monocytes are other than they probably look like this if you could see them up close. In any case, monocytes can differentiate into cells called macrophages. Macrophages have many functions, but the one I'm referring to here is the breaking down and recycling of red blood cells and the iron in them. If this didn't happen, we would need much more iron than we do. Macrophages do this through a process called phagocytosis, in which they engulf the entire red blood cell and essentially use enzymes to break it down into its component parts, including iron and amino acids. These are then recycled to be used in the process we've been describing of making new red blood cells. And here we have, finally, the red blood cell, which is collapsed into this final concave shape, a membranous sac containing not much else than 270 million hemoglobin molecules. Lacking a nucleus and other organelles, it can only last for 100 to 120 days before it needs to be replaced in the process I've just described. Now I'm going to shift gears a little bit and talk about two serious health problems which involve the hemoglobin in red blood cells. The first is diabetes and its precursor, prediabetes. The A1C test is often used to diagnose and evaluate diabetes and prediabetes. If you or a loved one suffers from diabetes or prediabetes, you're probably familiar with this test which a doctor may have prescribed. The hemoglobin in your red blood cells is subject to a process called glycation, in which glucose molecules, here's a glucose molecule, become chemically linked to hemoglobin molecules. It's normal to have some glucose attached to the hemoglobin, but the hemoglobin, of course, is inside the red blood cells, not on the outside, as these pictures suggest. These pictures depict red blood cells with hemoglobin and some glucose attached. The picture on the left would represent healthy red blood cells, and the one on the right, unhealthy amounts of glucose. The A1C test measures the concentration of glycated or glucose-filled hemoglobin in the blood. A normal reading for a healthy adult is 5.7%. Diabetes is often indicated by a concentration greater than 6.5%. For diabetes sufferers, the goal is usually to keep the A1C below 7%. The test must be repeated about quarterly because, as we've said, red blood cells and hemoglobin have a maximum lifespan of 4 months. The second health problem related to hemoglobin is sickle cell disease, or SCD, which is a group of inherited red blood cell disorders. In sickle cell disease, the hemoglobin is abnormal, causing some of the red blood cells to become hard, sticky, and deformed into a rigid sickle shape. And the sickle cells don't last as long as normal cells, causing a constant shortage of red blood cells. Symptoms include pain, anemia, swelling of extremities, infections, and stroke, as well as shortened lifespan. People born with one hemoglobin S gene and one hemoglobin A gene have sickle cell trait, or SCT. Usually, these people will have no signs of the disease. In this diagram, you see both parents were born with sickle cell trait. As you can see from the diagram, if they have a child, there's a 25% chance that the child will inherit one copy of hemoglobin A from each parent, and this child will be free from sickle cell disease. That would be this one here. No sickle cell disease because they both inherited hemoglobin A. There is a 50% chance, however, that the child will inherit one hemoglobin A and one hemoglobin S gene, in which case the child will also have sickle cell trait. So these two are like the parents. But there's also a 25% chance that the child will inherit both of the hemoglobin S genes, and in this case, the child will have sickle cell anemia, generally considered the worst form of sickle cell disease. In addition to sickle cell anemia, there are a few other types of sickle cell disease in which the child inherits one hemoglobin S gene and one of several other types of abnormal hemoglobin gene. This chart comes from the Centers for Disease Control and includes 5 facts about SCD. I won't go over it in detail, but the headlines are: first, a child gets SCD when he or she receives 2 sickle cell genes, one from each parent. Second, SCD has many faces, generally affecting people who come from the geographical regions shown in red, namely Central and South America, Sub-Saharan Africa, the Middle East, India, Pakistan, and Bangladesh. 3, SCD can be cured for certain patients. Fourth, anemia is a common effect of SCD, but it can be treated. 5, a person with SCD can live a long and high-quality life. For further information, you can check out the CDC's website. Let's talk about some life-saving aspects of red blood cells. Most of us over a certain age have been asked many times to donate blood to a blood bank. These donations are processed into what are called packed red blood cells, and they're used to save lives through blood transfusions for patients for various different causes, including traumatic injuries, surgeries, severe anemia, and therapy for certain cancers. This practice only became widespread in the 1960s, but is now almost taken for granted. According to the CDC, trauma is currently the leading cause of death for those under the age of 59, and transfusions are often the only life-saving intervention available for many trauma victims. Notice that this particular unit of blood is labeled type B. Human blood is categorized into four main groups: A, B, AB, and O, and each group can be either Rh positive or negative, meaning there are actually 8 groups. Each person has a unique type. Mine is A positive. The blood from each group has unique antigens and antibodies, and care must be taken to make sure that transfusions are compatible between donor and recipient. Group O red blood cells do not have any antigens, so Group O blood can generally be given safely to any other group. Group AB red blood cells have no antibodies, so someone with type AB blood can generally receive any of the other blood types. If you need a transfusion, your blood will be tested to make sure it is compatible with the donor's blood. But as I mentioned, it's always a challenge to get enough packed red blood cells to meet the needs of trauma victims worldwide. So is there a way to manufacture red blood cells without having to have people donate their blood? It turns out that there are companies working on that as we speak. And here's one. Icosia Biotechnology with their product erythrocin was one, unfortunately. Their product would have relied on genetic engineering techniques to manufacture human red blood cells from a source of donated stem cells. As we've seen, there are numerous steps from the stem cells to the finished red blood cells, so this has not been an easy task, and unfortunately, I can't find any current information to indicate that Icosia is still in business, unlike last fall when I checked and it was in business. Starting in 2019, they attempted to create a truly universal blood product so that healthcare professionals would never be limited by the availability of red blood cells and could concentrate their efforts on providing critical treatment. The Icosia product erythrocin is an example of biomimetics, which is using synthetic methods to mimic biochemical processes. In this case, Icosia planned to use human blood stem cells in a proprietary process involving genetic engineering and a manufacturing process which did not depend on bone marrow to create this product. Icosia, as I said, was founded in 2019, and I have no idea what's happened at Icosia, but they were still in business a year ago with optimistic projections. It's clear that biomimetic systems were created by human minds, but how much more likely is it that the biological systems upon which they were based were not the result of random, unguided evolutionary processes, but were created, in fact, by an intelligent mind? Fuzz Rana, in his book Thinking About Evolution, says, biomimetics naturally flows out of a creation model approach to biology. Using biological systems to inspire engineering makes better sense if the designs in nature arise from an intelligent mind. Now let's recap the 4 mysteries I have described. The 4 mysteries: how red blood cells know they must lose nucleus and organelles. 2, how they know to differentiate given they all have the same DNA. 3, how heme knows it must incorporate an iron atom. And 4, how hemoglobin knows it must incorporate the non-protein heme. Some scientists may discover mechanisms which explain these mysteries, but even then it will be very difficult to explain how those mechanisms could have come about through naturalistic processes such as evolution. Throughout this presentation, I've tried to make the case that just the one problem of how to make a red blood cell is extremely complex. But most of those who subscribe to the idea that all of life can trace its origins back to the first prokaryotic cell almost 4 billion years ago recognize that life is very complex. For instance, back in 2008, we have this quote: "The complexity of living organisms is staggering, and it is quite sobering to note that we currently lack even the tiniest hint of what the function might be for more than 10,000 of the proteins that have thus far been identified in the human genome. There are certainly enormous challenges ahead for the next generation of cell biologists, with no shortage of fascinating mysteries to solve. This was a quote from Molecular Biology of the Cell, 5th edition, page 142, by Bruce Alberts et al. In 2008. But here are some quotes from two other more prominent supporters of the evolutionary paradigm. Biology is the study of complicated things that give the appearance of having been designed for a purpose. Dawkins, of course, is one of the so-called new atheists, and Francis Crick was one of the discoveries of the DNA molecule back in 1953. Oh, I haven't read Crick's yet. Biologists must constantly keep in mind that what they see was not designed, but rather evolved. On the other hand, the Bible tells us in the first verse of the book of Genesis that in the beginning, God created the heavens and the earth. And in the first chapter of Genesis, we learn that God created the first plants, fish, birds, and mammals. Seems to me that whether you believe that all living creatures are descended from an evolutionary tree of life, or that God is ultimately the creator of the universe and all life, one needs to have a great deal of faith. We Christians, of course, have faith in God as the creator, but even if evolution is true, as we've pointed out in this presentation, scientists have no explanation for many things. For them to accept evolution as the best explanation for all the complexity of life, I think, also takes a great deal of faith. It's been my argument throughout this presentation that red blood cells did not evolve through random, unguided natural processes, but that they show unmistakable signs of having been created by a master designer, who I believe to be the God of the Bible. I believe the mysteries I have cited provide some evidence suggesting that processes or recipes are designed, supporting my contention. But beyond these signs, the Bible tells us that Christ gave his life and shed his blood to pay the debt we owe for our sinful natures. For in the Garden of Eden, Adam and Eve ate of the fruit that God had forbidden them, and by so doing, they brought sin into the world. And since then, every human being except Jesus himself has sinned. As Romans 3:23 says, all have sinned and fall short of the glory of God. But God chose to make provision for us sinful people to have a relationship with him who is without sin. In the days before Jesus came to earth, God established a practice of animal sacrifice, including the shedding of blood to atone for the sins of men. The Old Testament book of Leviticus says, "For the life of a creature is in the blood, and I have given it to you to make atonement for yourselves on the altar." It is the blood that makes atonement for one's life. And in the New Testament, without the shedding of blood, there is no forgiveness. Hebrews 9:22. But God sent his Son, Jesus Christ, to earth to be the final sacrifice to redeem us from sin. God presented him, that is Jesus, as a sacrifice of atonement through faith in his blood, as we're told in Romans 3:25. We're also told, "For God so loved the world that he gave his one and only Son, that whoever believes in him shall not perish, but have eternal life." John 3:16. In his systematic theology, Wayne Grudem tells us, only the blood of Christ, that is, his death, would be able really to take away sins. There was no other way for God to save us than for Christ to die in our place. In summary, we have thought about how red blood cells transport oxygen throughout our bodies and help to remove the excess carbon dioxide our bodies produce. We have recognized that unlike other cells in our bodies, red blood cells have lost their nucleus and other organelles, and so after 100 to 120 days, they must be replaced by a complex series of steps, which look designed. We've considered 4 mysterious processes, each of which appears designed, and we've looked at 2 diseases involving the hemoglobin in red blood cells and have seen the important life-saving role red blood cells play through blood transfusions. We've considered an example of biomimetics, even though it didn't seem to work. And finally, we have been reminded of the price Jesus had to pay on the cross by shedding his blood to redeem us from our sins. This concludes my presentation, and I'll be happy to take your questions. Okay.
I've got a question, Kirby.
Go ahead, Lothar.
Um, how is age related to the trillions of and millions?
Okay, so the question is, how is age related to the trillions of cells? Not apparently. There doesn't seem to be any correlation. I think it's more the size of the body that depends on how many cells there actually are. So,
I mean, are there kinetics involved that it slows down the formation and?
not that I have read. It seems like you've got 120 days to replace all those red blood cells, and 100 to 120 days, and you're just constantly cranking them out to replace the ones that are dying.
Good to know.
Yeah. Anybody else? Any other questions or concerned or
these different kinds of cells in the body? Are they the sizes? Are they
all? They're all different sizes.
They're
They're all
all the same size.
different sizes. That's yeah. They're all different sizes, and that's a great question. But yes, and we. What bothers me about the whole cell differentiation process is that so many texts that I have seen now seem to reiterate this idea that, well, it's all in the way these gene products differentiate, but it never specifies how those differentiated parts get into the different cells. It's just basically, well, the gene products do it, and I have to admit that the gene regulatory networks that control all these genes are very, very complex. And so you can almost nod your head and say, well, okay, well, that should do it, shouldn't it? Well, I don't think so. I think there's got to be something else. And I actually think that a good answer for this may be somewhere lying within the cytoskeleton in the cell. So each cell has something called a cytoskeleton in it, which basically establishes the polarity of the cell, the top and the bottom, and the dorsal and ventral, and all those different spaces. And I think somehow there's a good chance that the cytoskeleton will be involved somehow in the cell differentiation process when it's finally uncovered.
I have a question.
Yeah, go ahead, Bill.
You threw some big numbers at us, 22 trillion,
Yes.
30 trillion.
Right.
How is that stuff measured? How do I know there's 370 trillion helixes in a red blood cell?
That's a huge number. Yeah, I mean,
did you count them or what?
No, I'm depending on whoever did, and I'm sure that the way they did it was they took like a cubic centimeter or something of the body, or maybe even a cubic millimeter, and counted the cells in that, and even that is going to be pretty large, when you multiply that times all the cells in the body. But it was done something like that, and how precise that is, I don't know. I mean, I think it's like one significant figure accuracy myself, but when I gave 33 trillion or 27 trillion. I don't know, that's as close as they could come.
I mean,
Kirby, it looks like
these numbers is like talking about the universe.
You're right. And that's your body, my body. Belinda, you had a question.
Okay, just to summarize then, the first mystery, I have my note here. How does the our red blood cell know when to lose its organelles? Was one.
Yes.
Okay, and then the second was about how do the stem cells know when to differentiate to the different types of cells,
Yes,
and as well as the body plans, and then
Well,
currently,
not so much. I wasn't really specifically raising the body plan. I did discuss it, but that's not really part of the mysteries at this point.
oh, okay.
So that's, to me, that's another mystery too.
Oh, okay.
If you are an evolutionist, I think you have to say, well, evolution did it, so there is no body plan. It's just that because we evolved the way we did, we're humans. If we had
Mhm.
evolved differently, we'd have been Neanderthals. If we had evolved still differently, we would have been redwood trees. I don't
I
know,
see. But that the differentiation part, though, currently is probably more explained by the junk DNA when things are turned on and off.
Well, actually, Sean Carroll is the only one that I've heard anyway that said that it's contained in the DNA.
Mm.
Most evolutionary biologists will say, it's all within the genes that are there in the cell. Of course, they're all there. They're all in every cell, but some are turned on and turned off differently because of these gene regulatory networks. That's basically the explanation that we have now. But to me, that doesn't really answer the question. How do they differentiate? Because they all have the same DNA. So
Okay.
that's where I'm stuck, and if any of you can come up with the answer, I'd love it. But so far in all the books that I've been reading and all the articles that I've been reading, not that that's an exhaustive list, but at least I have been trying. I have
No,
not
thank you.
found an answer that satisfies me.
Okay, thanks, Doug. Kurt.
Go ahead.
I'll try to make it quick.
Yeah.
A matter of clarification. You mentioned epigenetics, and it seems like that's a real popular topic now, if I understand things right. And I'm trying to understand, is that epigenetics is those genes that are regulating the expression of genes that code for proteins. Is that correct? Or is it
Well,
machinery that exists
okay,
outside
so
of DNA?
I almost hesitate to mention epigenetics because I think that we really don't understand enough about epigenetics to be talking intelligently about it. I will say this, that what I know about epigenetics is it's that certain parts of the DNA have pieces of methyl attached to them, and that methyl, if it's attached to a cytosine, in a cytosine that's close to a guanine, can turn off the nearby gene. Similarly, DNA is twisted around histones, and the histones have acetyl groups that are attached to them, which turn on the genes. And we don't really understand exactly how those methyl groups, well, we understand how they get put on. They get put on by a particular enzyme, and as well as the acetyl gets put on by a certain enzyme. But why? Why do they get put on? And how is it that they specifically get put on? That's all part of the mystery as far as I'm concerned. And, and the very fact that we don't know that much about epigenetics, I just mention it because I happen to be interested in studying it myself. The fact is that I can't find very much about it that's really known.
I think there's a Wikipedia page on it, but isn't it also those things that are attached then get passed on if they are in?
They can, yes, they can.
Yeah, so
right.
it can be inherited.
However, as it turns out, when a new egg is formed, I have read anyway, that all of those epigenetic markers get wiped out. So exactly how they reform, as the body is being created, I can't answer that question.
So I was blown away by your one of your first slides, which said that 80% of the cells in our body are red blood cells. And I'm like, wow, that's crazy. What about the rest of us? Since we're, muscle and bone.
So we're only talking
so
maybe 10 trillion cells.
Mm-hmm.
That's quite a few cells.
So the muscle and bone are only 20% But now that's the
Well,
number of cells, though.
yeah, but
So
Are they bigger or?
probably bigger.
I guess I'm just wondering what other kind of cells are there roughly besides the blood cells?
oh, there's
that
there's
make
like
the
there's like
20%?
200 cells. So for example,
Oh, okay.
there's liver cells, there's kidney cells, there's pancreas cells, there's lung cells, there's skin cells, and
But
there's neurons. Neurons are also cells.
sure, but I
Mhm.
think you were saying that the liver, no, from the stem cells, some of
Yes.
them become liver and so on. All right, wow, that's crazy. Thank you.
Or who?
So most cells are taking in nutrients and doing work and stuff like that. But it sounds like that all depends on the nucleus and the organelles and whatnot. And so red blood cells don't consume any nutrients. Is that correct?
Correct. That's right. They live for 100 to 120 days and then they die.
Or so are they really living if they aren't consuming?
Great question. That's a lovely question. I would say no, but the fact that they're doing useful work to make us live,
Yeah.
because I like that question though. They do not have DNA in them, so and I think that kind of technically says that they're not alive, but
It's kind of the flip side of a virus, which has DNA but doesn't do
yeah.
anything on its
Well,
own.
it has most of them have RNA, not DNA, but
Oh, okay.
yeah. Good question. I like that question. Anyone
So
else?
along that same line, I mean,
Yeah.
being able to make red blood cells from just chemistry, is this possible? Or do you need red blood cells to make red blood cells?
No, you don't need red blood cells. I mean, you need a blood stem cell, and then you need a lot of ingredients. And I guess as Icosia found out, it's not as easy as it may look to simulate, the process that we have naturally in our bone marrow. They tried to build a tank that would do the same thing that the red blood cell is doing, but for some reason, I don't know why, I don't know what it was that didn't work for them, but apparently it didn't, and I guess they lost funding or something. I wouldn't be surprised if that they may at some point resurrect, but I don't know that for sure. So
Okay.
yes, Wu family has a hand up.
Yeah, I was wondering, since you said that the red blood cells don't have DNA,
Yeah.
does that make the Jurassic Park, like the plot, does that make it impossible?
I guess I don't follow that.
Jurassic Park. Jurassic Park, the movie where they
The
Oh,
movies, like
okay.
extracting the DNA from the blood, yeah.
Oh, since I didn't see Jurassic Park, I guess I'm kind of at a disadvantage. I think that what they did in Jurassic Park was to somehow manage to capture some DNA and then create a dinosaur.
Yeah, they got like a mosquito that had drank the or gotten the blood from a dinosaur, yeah.
No, I would say that I think that's a good question. Frankly, I'm not sure I'm qualified to answer it. Especially since that it doesn't sound like to me that the whole basic premise of Jurassic Park works, because DNA doesn't really last that long. But let's see. So if you could pull together, yeah, I guess if you could pull together some sort of proxy of managing to put together a clone, maybe somehow, I think that you would be able to create the red blood cells again from using, you can create the stem cell. If you can create the stem cell, you should be able to create everything else. So no, I think that the premise I don't think precludes being able to create a dinosaur if you were able to actually create or to have a sample of dinosaur blood. which I
But
don't.
you would have to go with a different cell other than the red blood cell then. Do the white blood cells or the other monocytes and whatnot, do those contain DNA?
Sure, yeah.
Okay,
Yeah,
so
the only one that doesn't is the red blood cell.
okay, so if they grab that out of the
But the
blood,
point is that stem cell exists. Remember, stem cells can reproduce themselves, and the
right, but
blood stem cell is still there. So, and which is thank goodness, because if it wasn't, we wouldn't be able to replace our red blood cells.
Thank you.
Thank you for the question. Anybody else?
Okay, so I've got another one, Kirby.
Yeah, go for it, Kofi.
The ability to carry a lot of oxygen in the cells. That is, I think you pointed out that this is actually due to that iron, right?
Yep,
That
yep.
iron, right?
Yes.
Is there is that optimal, right? Is there some other atom that could have been incorporated?
That's a great question too. All we know about is iron. There are a lot of other. Actually, this is one of my favorite topics, which is the incorporation of metals into proteins. This is a function of a subtopic of biology called biological inorganic chemistry, and you have all sorts of books. For example, let's see, I've got one somewhere here. No, I can't see it off the top of my head, so I must have hid it away. But something called metalloproteins, and and there are a lot of. I mean, iron is only one of many elements that are metals that make many enzymes. I'd say there's close to half of the enzymes have some metal of some sort in them. And it can be iron, it can be molybdenum, it can be cobalt, can be vanadium. I'm working on a paper right now, which is talking about how nitrogen is broken down in. It's actually only bacteria can do this. Plants, animals can't do it. They have to have bacteria working with them to break down nitrogen. And they do that through an enzyme called nitrogenase, which incorporates molybdenum, of all things. And if there's not enough molybdenum present, instead they'll use vanadium. And if there's not enough vanadium present, they'll use iron. Well, molybdenum seems to be optimum. So is in this case, is iron optimum for red blood cells?
Yes.
Seems like it might very well be. Yeah.
Okay, because that's, of course, there's a sort of apologetic value there as well, right? Why is it that apparently that sort of the optimum of all these other metals that could have been, is there something about iron that makes it easier to function in a cell, or is it there simply because of its ability to allow the cell to absorb more oxygen?
That's a great question. I think beyond my knowledge band, I guess if I had a better knowledge of chemistry, I might be able to answer that question a little better. But I think it's almost self-evident that it's true, but I can't say why.
Okay. Yeah.
Sorry. Anyone else? Any other questions? I love these questions. You guys got some good ones here.
Yeah, I mean these comments or questions that have to do with optimality. These are the ones that often interest
Right.
me.
Yeah.
Right.
Yeah.
You see this again in the genetic code, right? This mapping of these, right? You have these 20 or so proteins, and you have 64 possibilities of these bases.
Right.
And the mapping from the bases to the proteins is apparently optimal, right? In terms of so yes.
Getting back to the incorporation of metals in proteins, though.
Yes.
That one to me is a head scratcher. What is it that causes a particular enzyme to incorporate iron or molybdenum or whatever?
Yes.
Iron often in this case, it's just iron all by itself, but iron often is incorporated along with sulfur molecules. So you might have 2 irons and 2 sulfurs, or 7 irons and 8 sulfurs, or various different combinations of sulfur molecules, both of which are pretty common in some of the electron transport chains in both mitochondria and chloroplasts. So, and as I said, that's one of the options, not as efficient as molybdenum in the nitrogenase that I mentioned, but it's also a possibility. So, okay. Sorry, I don't have answers to all of these questions, these great questions, but anyone else want to try for one?
This is not quite a question, it's more a wondering. I heard you say, Kirby, that the red blood cells are recycled, and that if they weren't, we would need a whole lot more of them. Could you just say a little more?
The numbers kind of speak for themselves. I think if we have 33 trillion or 33 trillion cells in our bodies, and 27 of them are 27 trillion are red blood cells. Kind of getting back to Bill Mignot's question, I think, which is that the numbers are staggering. They're so ridiculously large as to be mind-boggling. But yeah, I mean, if all 27 million of them just died, we didn't have any way to reproduce them, that would be a big deal. That's a lot of iron. That's a lot of protein pieces, which have to be reconstituted somehow. So just to have them broken down and fed back into the bone marrow is a good solution to basically how it works. That's the way I see it. I don't know if that helps or not, but does that make sense?
Yeah, it's kind of on a rolling basis then.
Yeah, right.
Right, but that way you don't have to eat that much iron every day.
Exactly.
To
Yeah.
generate the trillions of cells, whereas if you have recycling, you don't have to eat nearly as much iron.
Exactly. Yeah. And that's just one of many other
pieces.
I don't know about the vitamin B12 and folic acid, whether they become part of the breakdown process of red blood cells or not, but
Kirby, I like your addition of Vazrana's quote, just to kind of support your idea that this recipe speaks of design and such, and just that, and then kind of what Coffee is also mentioning about there's engineering type of optimization
right.
happening as well. And then because of that, that's how that biomimetics, if you have this biological systems that show the intelligence, required, like what human engineers do in terms of modeling, that there's an intelligent mind behind the design of this biological system, the red blood cell.
Yeah. One example that I can think of that, two examples. One is the failed Icosia example, which taking a process that seems like it's should be kind of simple, turned out not to be so simple. And reminds me also of Craig Venter, who is one of the fellows who helped decode the human genome in the first place. And then after that, he decided he was going to create a special genome of a prokaryote, of a bacterial model. So he took a very simple bacteria and tried to create this DNA for it. And it took him several years with several different PhD experts working on it. This is kind of the fuzz rana idea that even this kind of a model, even though it didn't actually, he never was quite able to do what he was trying to do, but he eventually created something. He created something that was not the original prokaryotic cell. It was a somewhat slightly different cell. So he claimed victory for that, even though he didn't succeed in what he was originally trying to do. But just shows how difficult even the simplest, obvious type problem can be. I guess one third example, I'm Karen and I have just recovered from COVID, and but it makes me think of the development of the vaccination, the COVID vaccination, which is basically taking mRNA, creating the mRNA to create the protein from the COVID vaccination, and how this fairly simple idea took years and years to actually develop and come up with a solution that worked. And the woman who won the Nobel Prize this last year for doing that, Kariko, her last name was Kariko. Can't think of the first name, but she'd been working on that for 20 years before they had a final breakthrough that enabled them to develop that mRNA vaccination. So these are difficult problems. Biomimetics is not as easy as it looks or sounds.
Mhm.
You got to hold your mouth just right for it all to work out right. Okay, well, any other last minute questions? We could keep on going for a long time here, but I think let's let me close this meeting with a word of prayer, and we'll if anybody wants to stick around afterwards and chat for a while, that's fine with me. And Karen and I will be here for a little while. But let me close this meeting with a word of prayer. Gracious Lord God, thank you so much for the opportunity we've had to be together tonight, to learn more about this remarkable little red cell in our bodies that is full of life and full of the life that you created for us. We just praise you, Lord, for your miraculous creation. We ask you to give us wisdom as we go through our lives, and please bless this time we've had together. Go before us as we start our a new chapter in our lives every day. In Jesus' name we pray. Amen.